Several years ago, the Lake Michigan Committee formed a task group to look at the, we were called the Native Planktivore Task Group. Here's the members here on the screen. The objective of that was to actually look at the scope the feasibility of restoring native planktivores, concentrating mostly on deep water ciscos and Coregonus artidi, and develop a white paper. We went ahead and developed quite an extensive white paper that we distributed last year. It's still in draft form. What we did is, as a committee, so the strategies for recovery, we talked about a lot of things in that report. We talked about impediments and challenges, opportunities. The strategies for recovery were kind of outlined like this. Certainly protect remnant populations of loader and cisco, and more importantly, and what we've been hearing about is reintroductions of extirpated species or species that are very low abundance. So our major impediment really is we just don't have any fish. So, and habitat improvement was also in the mix there, but likely not going to certainly affect things the way reintroductions would. So the ecological goal here is to provide forage to piscivores, reestablish the vertical and horizontal energy transfer across habitats and trophic levels, something that was disrupted. And the short and long-term objective was to provide a self-sustaining shallow and deep water native forage base that can use the existing invertebrate resources, which have changed over time, but are, even though we've seen a truncation in the size composition of zooplankton, we've seen diaporia disappear, we still appear to have a zooplankton community that's very similar to Lake Superior, both in species composition, size composition, and they actually support Coregonians there. So that's the logic behind that. Obviously, the apical biomass we could probably support with the current zooplankton community is much lower than it was historically, but it's not an impediment, we feel, to get something going, especially for reintroducing these species. So the longer term objective would be obviously to provide fisheries on populations with surplus yield, but really what we're trying to do is get populations established, self-sustaining to feed fish, predator fish. So looking at a continuum of difficulty for reestablishing these animals, cisco, Coregonus artidi would probably be the least difficult. Short jaw and blackfin cisco, the hardest, just by virtue of the fact that their available source populations are very, very limited and hard to get at. Lots of kai out in Lake Superior, probably weather and those kind of logistics would prevent some difficulties. So where the committee ended up kind of coming to a standstill was the idea when we started talking about source populations for artidi. And there was a lot of discussion and about whether we would stick with the small remnant populations of artidi in Lake Michigan, which are small, or could we go outside Lake Michigan and hit up some of the larger source populations that we see in Lake Superior. We couldn't come to an agreement, so the idea was to task a group of geneticists that are very familiar with Coregonian genetics and ask them a series of questions that centered around genetic diversity, population size, genetic differentiation, real targeted questions on best donor populations, and what secondary sources. So we actually put together, invited these folks to actually answer that series of questions. Wendy Stott provided an extensive array of genetic information on a variety of populations, of RTD populations all over the Great Lakes. That was made available to the panel, and then the panel looked at that data and provided independent responses to our 5 questions. Those responses, I just got the last one from Brian Sloss. Everybody else on the list responded, with the exception, I'm still hoping Chris Wilson's going to provide something, but I can't seem to get a hold of him. And Jim Reese wasn't able to do it just because of health issues. So anyway, we feel the best donor populations look something like this. They have high genetic diversity, are from the same habitat type as where they're being introduced, obviously present no disease risks, are large enough to support routine gamete collections, and are certainly logistically and economically feasible. We plan on consolidating all the responses from the geneticists. I've asked Meredith Bartron from Fish and Wildlife Service and Chuck Kruger to assist me with this. Then we'll get it back out, all the responses back out to the committee, and prepare a response to the Lake Michigan Committee on this important issue.